Today we're continuing the story of ACE inhibitors. In the last episode, we uncovered the RAAS system. This revealed that the kidney isn't just a filter, it's a regulator capable of raising blood pressure through a powerful chain of enzymes and hormones. But once scientists understood how the system raises blood pressure, a much bigger question appeared. Could that system be safely turned off? The clue that helped answer that question came from an unexpected place: the venom of a Brazilian pit viper whose bite causes dramatic drops in blood pressure. So in this episode, we explore how did researchers create ACE inhibitors and how did those drugs treat not only hypertension, but heart failure and kidney disease too. And along the way, we'll also hear from a special guest, a cold-blooded Brazilian pit viper with a very strong opinion about pharmaceutical research. Welcome to the next episode of Mechanism of Action. I'm your host, Adam J. Brown, and we are digging into the second episode of the ACE inhibitor story. And just a quick recap of the first episode, we talked about the work that it took to make the link between the kidneys and hypertension. For example, grinding up kidneys and then injecting the kidney extract into dogs and humans to see what would happen, and it would raise blood pressure. And eventually it was found that the mechanism of how the kidney regulated blood pressure involved multiple different enzymes and proteins interacting with each other. So these enzymes are renin, angiotensin, angiotensinogen, angiotensin 1 versus angiotensin 2. And it was discovered this angiotensin 2 was actually the culprit here. Angiotensin 2 is actually acting upon blood vessels and triggering vasoconstriction. And what was discovered was something called ACE, angiotensin-converting enzyme, which is what converts angiotensin 1 to angiotensin 2. So if we can block the conversion of angiotensin 1 to angiotensin 2, that could be a useful therapeutic. That's where we ended the first episode with that discovery. Today in episode 2, we are focusing on the first ACE inhibitors and the first clinical application of these therapeutics and how they changed the world of hypertension. But not only that, they actually changed the world of heart failure, and they changed the world of diabetic kidney nephropathy, as well as chronic kidney disease. And then we'll talk about some of the early experiments that led to our understanding and appreciation of how the heart failure could be involved and how chronic kidney disease can also be ameliorated with ACE inhibitors. Let's get back to that snake venom, the Brazilian pit viper. That was a deadly snake that lived in the fields, and when it bit, it would cause a person's blood pressure drop so profoundly that it caused hyperperfusion. We didn't know how it happened. In the late 1800s, there was a lot of interest in allergy. There was this question of, is there this allergic component to the body's response to snake bites? One of the biggest players of allergies at that time was histamine. It was shown to cause vasodilation. So histamine became a major suspect. Maybe histamine is actually triggering all of these responses to venoms or toxins. And eventually the Brazilian pit viper began to be investigated. A Brazilian doctor by the name of Mauricio Rocha e Silva was working on histamine and anaphylaxis when another doctor brought him a sample of the venom of the Brazilian pit viper, as he knew that the Silva was interested in venoms and histamine. At that time, the venom was known to cause blood clotting. Scientists wondered, well, maybe the blood clotting was somehow causing the shock. For example, having huge thrombosis like pulmonary embolisms and causing death. So they tested this idea on an animal model that had no fibrin or no clotting proteins, i.e. the animal could not form blood clots. They injected the venom into that animal and the animal still died. So they recognized that the hypotension and the vascular collapse was independent of blood clotting. So then they injected the venom into a dog and no increase of histamine was detected. What could this be? It was not histamine. It doesn't seem to be clotting. So what is driving this vascular collapse in people who get bit by Brazilian pit vipers? And another doctor in the same lab tested the venom on the ileum, or the gut tissue, that was pre-treated with antihistamine and atropine to block another known kinetic compound, acetylcholine. Much to everyone's surprise, the snake venom still produced an effect in the gut muscle contracted, independent of histamine, independent of blood clotting, independent of acetylcholine. So the snake venom was breaking all the venom rules. They did not know how it was causing vasodilation. So many of the venoms had fallen into one of these categories, but the Brazilian pit viper was not doing this. One thing that was noted was histamine and acetylcholine. Both of those caused prolonged and rapid onset contraction of the gut, whereas the snake venom produced a very slow-acting but steady contraction of the gut. Dr. Silva named whatever was in this snake venom brady, i.e. slow, and kinin, movement, so slow movement. Bradykinin was induced with this snake venom, okay? And Now we turn to the laboratory of Dr. John Vane in England in the 1960s, who was looking to understand angiotensin II. This is after ACE was described by Dr. Leonard Skeggs in Cleveland in the late 1950s. Dr. John Vane, who was a big deal, he had discovered prostaglandins in the laboratory. He's also instrumental in learning the mechanism of action of aspirin. His lab was studying hypertension and looking at different mechanisms of hypertension when a doctor from another country comes to join Dr. Vane's lab. This outside doctor happens to be Dr. Sergio Ferreira from Brazil. He brings venom from a Brazilian pit viper. Dr. Vane's lab was working on angiotensin II and trying to figure out how to block it via ACE, which was discovered a decade before. So they say, hey, can we see what the snake venom does to ACE, or angiotensin-converting enzyme? And lo and behold, the snake venom inhibited angiotensin I from converting to angiotensin II, thus inhibiting the hypertensive aspect of the whole RAAS system. So with these medical stories, we often focus on the scientists, the discoveries, the breakthroughs in medicine, but we rarely stop to think about the animals that were part of that journey. And it got me wondering, has anyone ever gone back and asked for the animal side of the story? Well, it turns out someone actually did. By sheer coincidence, it lines up perfectly with today's episode. An investigative journalist apparently tracked down one of the key figures in this whole ACE inhibitor saga, a Brazilian pit viper, and conducted an interview back in 1990. So, in the interest of balanced historical reporting, here is that interview.
Please note that voices in this interview have been digitally altered to protect anonymity. Tonight we're joined by Source J, an anonymous Brazilian pit viper who claims Big Pharma stole his venom and turned it into life-saving medication.
First of all, stop calling it life-saving. It's humiliating.
Right, sorry. Let me start over. You're considered one of the deadliest snakes in South America, and your venom is uniquely dangerous, correct?
Absolutely. We didn't just go with basic neurotoxins like some lazy rattlesnake. No, we engineered bradykinin-potentiating peptides, catastrophic vasodilation, a massive collapse in blood pressure. Elegant, lethal, it's art.
But scientists eventually realized that mechanism could be used to treat hypertension, which led to the development of ACE inhibitors.
Right. They took our proprietary murder juice, diluted it, put it in a little orange bottle, and now humans take it every morning with oatmeal.
That must be rewarding.
Rewarding? I barely leave my rock anymore. My reputation as a merciless, cold-blooded killer is in shambles.
I imagine it's difficult.
Difficult? It's textbook libel. They have caused irreparable harm to my brand equity. My personal brand is sudden, agonizing death. But because of their PR campaigns, when I slither into a village, people don't run screaming anymore. They politely step aside and thank me for their grandfather's improved cardiovascular health.
Have any other predators faced this issue?
Oh, the Gila monsters are furious. They spent millions of years evolving venom, and now everyone just associates them with rich housewives losing weight on GLP-1 agonists. It's humiliating.
Source J, thank you for sharing your perspective. Our listeners, many of whom prescribe ACE inhibitors every day, will definitely find this fascinating.
Prescribe?
Yes, a lot of cardiologists listen to the show.
Wait, you said this was an interview about my victimization.
Well, technically the episode is about the history of antihypertensives.
This is a medical podcast, isn't it? For sake!
Ah! No! Don't bite! Don't bite! Ah!
Remember this when you're on rounds, ACE inhibitors comes from a Brazilian pit viper. Isn't that pretty cool? The clear application of this pit viper, bradykinin-potentiating discovery, was we could potentially cause lowering blood pressure effect from doing this. Was there a rush of interest from pharmaceutical agents? No, not really. Again, this is the 1960s, and we still hadn't really fully grasped the problem of hypertension. The cultural shift had not really happened yet. There's also the additional problem of the drug was snake venom. It was designed to be injected via terrifying fangs. Patients were found to be less than enthusiastic to have fangs at home laced with moderate doses of Brazilian pit viper venom, even if the dose was moderate. And then if you swallow snake venom, it just degrades in your stomach. So you need to figure out how to make it a digestible pill. There's a lot of work to do. It wasn't just, hey, let's extract all this snake venom and give it to people and treat their blood pressure. So now you have to design a drug. Between 1970 and 1973, pharmaceutical companies randomly tested 2,000 chemical structures trying to design something that had ACE inhibition activity and was orally digestible. Okay. Scientists got a rush when they stumbled across carboxypeptidase A, which had some ACE activity. So they made 60 compounds similar to it, and within 18 months, voila, captopril was discovered. Captopril launched multiple randomized placebo-controlled trials. In 1982, multiple trials were published: captopril versus placebo, captopril versus hydrochlorothiazide, captopril plus hydrochlorothiazide, all demonstrating blood pressure reduction, and the combination with hydrochlorothiazide gave the greatest reduction. A brand new therapeutic option for hypertension was born. Of note, the initial dose tried was much, much higher than used today. Today, it's like 25 milligrams, 2 or 3 times a day of captopril. Back in the days, they used 100 milligrams multiple times a day, and people got hypertensive and passed out. That's not good. So captopril, you've probably heard of it, but you probably may have not used it. It has some niche uses, including scleroderma renal crisis, a rare complication of scleroderma. But you may be more familiar with lisinopril. How do we get to the more famous lisinopril? It actually has to do with making use of binding sites on the enzyme. Apparently, when looking at the binding of captopril to angiotensin-converting enzyme, it was recognized that not all potential binding sites were being occupied, and that, my friends, smells like opportunity. When did a pharmaceutical company ever turn down a potential binding site? I don't know of one. In 1985, enalapril came out soon after with an easier dosing, but similar to captopril, the initial dose was too high. Later, we realized we could use much lower doses and be just as effective. Then Merck took a stab at those binding sites, systematically altering each unit of enalapril, but substituting various amino acids. Eventually, lysine was found to be quite binding. What a binder. And then, because it was named lysine, they wanted to use the word L, and then we birthed lisinopril, which is once a day dosing, which took over the market. Now we're going to jump into the non-blood pressure indications of the use of ACE inhibitors. It turns out the RAAS system is not just important for blood pressure, it's in fact regulating a whole lot of different things. ACE inhibitors in this whole pathway are not used only to treat hypertension, but in fact they're utilized for a relatively wide range of indications. We're going to primarily focus on heart failure and diabetic nephropathy, i.e. kidney disease from diabetes, as well as chronic kidney disease not related to diabetes. So let's start with heart failure. What is heart failure? There's a lot of different ways of defining heart failure, but we're going to keep a pretty basic version of it, mostly talking about the heart not pumping as well as it should. So right-sided heart failure can happen when the side of the heart receiving blood comes in before it gets pumped to the lungs. And the left-sided heart failure is where the side of the heart is pumping out to the whole body. Left-sided heart failure is probably the most famous one, where the heart is not pumping as well. And then when that happens, that blood, instead of being pumped from the heart, slowly starts getting backed up. The heart pumping could be so feeble that the blood pressure is actually low in patients because the heart just can't provide enough oomph to actually raise the blood pressure. This could also lead to kidney issues. Heart failure notoriously causes kidney problems because the kidneys are being poorly perfused. And what happens when the kidneys are poorly perfused? We all know the kidneys start pumping out renin, which then knocks this whole RAAS system in place, and then you get vasoconstriction. So it can hurt the kidneys, hurt the lungs, and hurt a lot of different things when the heart is not pumping well enough. What was the treatment landscape of heart failure when ACE inhibitors were starting to come around? Mercury was used well before the 1950s to cause diuresis before we get hydrochlorothiazide. Digoxin was used very commonly in heart failure. It was thought to increase contractility of the heart. That's a whole nother story. Then we had vasodilators like hydralazine were used to decrease afterload pressure of the heart, which is what we think about as peripheral vascular resistance. So when the heart has to pump, if the pressure throughout the whole body is high, the heart has to pump even harder to go against that increased pressure throughout the body. Hydralazine was used to open up the blood vessels and decrease the blood pressure so the heart does not have to pump so hard. Beta blockers are a famous addition to congestive heart failure, similar around the timing of ACE inhibitors, but ACE inhibition was used in heart failure before beta blockers. In 1960s, 1970s, there was a recognition of the RAAS system being activated in patients with heart failure. Remember, renin is the hall monitor of the kidneys. It's monitoring whether there is too much or too little blood flow. When the heart isn't pumping well, there must be too little blood flow, which will increase renin levels. So not surprisingly, patients with heart failure often have elevated renin levels. They found elevated renin most consistent in right-sided heart failure, and the right-sided heart failure was also the most consistent animal model used, and that was actually found to have strikingly high levels of renin. And for this animal model, there's a dog. They opened up the chest, clamped the pulmonary artery, and that caused a fixed outflow obstruction, and that increased right ventricular afterload, increasing the systemic venous pressure. So when the heart's not pumping well, it seems to activate renin production. A quick little aside here. I was surprised to see that right-sided heart failure was the animal model that was used most consistently for this, because I think about heart failure most commonly with left-sided heart failure, that part of the heart pumping into the systemic circulation. The reason that was is because that left-sided animal model would cause death pretty quickly. Because with the left side, it would clamp the aorta, the main vessel going out into the rest of your body. It turns out if you did that, even just a little bit, it would cause arrhythmias. The heart would actually start beating inappropriately, and it would cause mass hypotension, and the animals would die. Sometimes they would live a little while, but it was very inconsistent. So one of the problems with this left-sided heart model was that the animal often wouldn't live very long. So that got me wondering, what was the first left-sided heart animal model? It turned out it was the coronary artery ligation model. What does that mean? Well, they would do a left thoracotomy, so open up the left side of the chest of a dog, and then go in and cut the left anterior descending coronary vessel. So it's one of the biggest blood vessels of the heart. And that would cause immediate death to the myocardium, leading to an immediate fall in left ventricular contractility, would tend to cause arrhythmias, and the size of infarct, they have variable amounts of damage. So it wasn't that consistent. But apparently it was more consistent than clamping the aorta. Now back to the point of this whole episode is, what about ACE? As mentioned before, patients with heart failure seem to produce a negative feedback, driving up renin, which is driving up angiotensin 2, which is causing vasoconstriction, which is causing more systemic vascular resistance, which is causing the heart to pump harder. So this is not just a pump problem where the heart is just not pumping well enough. It's this neurohormonal problem where it's actually releasing these other hormones and the enzymes in the body that are perpetuating this negative process of heart failure. So some early studies looked at captopril, the ACE inhibitor, and demonstrated objective measures like reducing afterload, improving cardiac function, and increasing exercise tolerance in patients. Captopril gave a hint in 1970 that, hey, this may actually have some beneficial effects in patients with heart failure. In 1987, we got a publication of the Cooperative North Scandinavian Enalapril study. It's called the Consensus study. This is looking at 253 patients and the average EF of 20%, so it's pretty bad, pretty low ejection fraction. Normal ejection fraction is like 45, 55%. So these patients are often very symptomatic. Around this time, in the 1980s, the mortality rate with someone with the EF that low was about 1 year. 50% of people with heart failure would die within 1 year. So we've had tremendous improvement in quality of life and mortality since these studies. It gives you an appreciation of how far we've come. In this study, the 1 year mortality in the placebo was 52% and the enalapril was 36%, so significantly lower at 1 year in the enalapril group. The number needed to treat to prevent death at 1 year in this trial was 6, which is pretty good. The trial was actually stopped early because of its clear survival benefits. The fun anecdote to this is all the trialists were really upset that the trial was stopped early. These people were blinded, randomized double-blind controls trial, and the trialists who were given the medication didn't know if they're given the placebo or the actual drug, the ACE inhibitor. And when they found out the trial was being stopped, they were so disappointed that was being stopped because either it was causing harm or the trial wasn't worth doing anymore. But then when the pharmaceutical company presented the data on the survival effect and the impact on quality of life, apparently there was a quote-unquote spellbound silence. They'd never seen a medication demonstrate such a morbidity and mortality benefit before in heart failure. And this validated the neurohumoral hypothesis of heart failure. Heart failure is not merely a pump problem, but also a maladaptive RAAS activation problem. Nice going, RAAS. A couple little more things. In 1992, the New England Journal of Medicine produced the survival and ventricular enlargement trial, the SAVE trial, great name, reporting a 20% reduction in mortality in heart attack patients given captopril. Another study, studies of left ventricular dysfunction trial, the SOLVED trial, showed 29% reduction in patients with asymptomatic left ventricular dysfunction. As you can see, in the 90s, there's a big deal for ACE inhibitors and the changing landscape of treating heart failure. Now we're going to switch gears from the heart to the kidneys, which are sort of the culprit in this whole problem anyway, because the kidneys are the one releasing renin, which causes all these problems. I'm not pointing fingers to the nephrologist. I'm not pointing fingers. So we're talking about diabetic kidney disease and chronic kidney disease. Patients with diabetes often led to diabetic kidney disease. Patients with high blood sugars over time can actually cause damage to the kidneys, which leads to the kidneys excreting lots of protein. Proteins are this marker that you can judge the degree of kidney damage based on the kidney function, but also the degree of proteinuria. Before 1980, the natural history of diabetic nephropathy was considered progressive. Basically, it's just going to slowly keep going. In patients with proteinuria and who had diabetes, 60 to 80% of patients would progress to end-stage renal disease in 10 to 15 years. So the majority of patients would be on dialysis in 10 to 15 years if they had diabetes and if they had proteinuria. Again, this is back in the day when trying your best to control diabetes was by controlling the sugars, because there wasn't a lot of other mechanisms to treat or decrease the impact of the damage of diabetes on the kidney. So diabetes wasn't the only thing that was causing kidney disease. Patients with elevated blood pressure would often develop what's called chronic kidney disease, and that would often be progressive. This condition was a little bit more malleable. If patients got control of blood pressure, the chronic kidney disease would decrease. In general, if it was caused from hypertension. Why do we use ACE inhibitors for these conditions? To answer that, we start with the Munich Wistar rat, which was a rat mouse model that was used to understand the pathophysiology of chronic kidney disease and diabetic nephropathy. Why was this rat used? At baseline, it has less glomeruli, the filtrating part of the kidney, and is prone naturally to chronic kidney disease and proteinuria within the first few years of its life. But also very conveniently, it actually has the glomeruli on the outside of the kidney. If you open up the rat's belly, you can visualize the glomeruli on the outside of the kidney. I didn't know that. Isn't that really interesting? I had no idea. So that was the Munich Wistar rat, was a great model for chronic kidney disease. Dr. Barry Brenner out of Harvard took advantage of being able to visualize these little tiny glomeruli. And Dr. Brenner said, I'm going to measure pressure within the glomeruli. And people gasped, that's impossible. These little rat glomeruli are super small. How are you going to measure in a rat? Well, it turns out he developed really, really small glass micropipettes and very meticulously inserted them into the glomeruli on the capsule of these Munich Wistar rats. And in doing this, his group demonstrated that rats with chronic kidney disease had highly elevated intraglomerular pressure. Remember, the glomeruli has the afferent arterial, the blood vessel going into the glomeruli, then the blood gets filtered. Then you have the efferent arterial, and that's a part of the vessel leaving the glomeruli. What was happening was he actually found that the efferent arterial in the rats with diabetes or the chronic kidney disease actually had narrowed. So all that pressure going to glomeruli couldn't be released, so the intraglomerular pressure spiked. What was driving this narrowing of the efferent vessel? Drum roll. Turns out that angiotensin-2 was doing this. The culprit again, angiotensin-2. The kidney is doing it to itself. The kidney is the one excreting the renin, causing this whole problem. Nice going, kidney. It turned out administering ACE inhibitors rapidly dilated the efferent arterial opening, and that popped open the efferent vessel, which rapidly dropped the intraglomerular pressure. They started giving ACE inhibitors to the rats. And lo and behold, the proteinuria decreased and the kidney disease progression decreased. Was it just because we're low on the blood pressure? So they used other blood pressure medications like hydrochlorothiazide, which also decreased the blood pressure, but it did not decrease the intraglomerular pressure, and it did not decrease the proteinuria. ACE inhibitors are actually decreasing the proteinuria and the pressure of chronic kidney disease, independent of their ability to lower the blood pressure. That's the big takeaway. They're doing something different than just lowering blood pressure that was saving the kidneys. Then we get the trials. The first major diabetic trial was the collaborative study group captopril in 1993. It was in type 1 diabetes, showing reduced creatinine over time, reduced progression in end-stage renal disease, and reduced death. I was surprised that type 1 diabetes was evaluated first, but it turns out they were trying to use the most consistent homogeneous group because they recognized type 1 diabetes didn't have all the atherosclerotic risk factors, so they didn't have other compounding problems that could make the data more difficult to interpret. Type 1 diabetes time course was also more predictable, requiring a smaller sample size and shorter follow-ups, etc. That was diabetic nephropathy. So what about chronic kidney disease not from diabetes, like hypertension? So Dr. Brennan also investigated that and also found the efferent arterial to be narrowed and increasing intraglomerular pressure. So they gave ACE inhibitors to those animals and it helped. Then they did the same thing with humans, and it showed that patients with chronic kidney disease from other causes from diabetes also benefited from these ACE inhibitors. The APRI trial, for example, was published in the New England Journal of Medicine in 1996. Chronic kidney disease from diabetics and non-diabetic causes. 583 patients followed for 3 years, baseline creatinine 1.5 to 4.0. Most patients were hypertensive. Benazepril significantly reduced the risk of renal disease progression, 50% relative risk reduction across different causes of chronic kidney disease. Also produced a substantial reduction in proteinuria. Clearly demonstrated lowering proteinuria is renal protective. These trials completely changed how we look at chronic kidney disease and diabetes and non-diabetics, both from a pathophysiology standpoint, but also from a treatment standpoints. We went through a lot on this episode. We discovered why snake venom was causing people to pass out in the fields and potentially dying in the fields was because they were so severely inhibiting ACE and dropping angiotensin 2 levels and causing hypertension. Once they figured that out, they started developing molecules that can be swallowed, and they developed captopril. Captopril was the first medication used that eventually led to enalapril and eventually led to lisinopril. And all these medications were shown to clearly have blood pressure lowering effect that was pretty safe and well tolerated as long as they didn't use whopping doses of it like they did back in the day. Then, as we started understanding this RAAS system, this neural hormonal maladaptive response to different diseases, they recognized it in patients with both right-sided and left-sided heart failure, and recognized captopril, enalapril, and lisinopril can reduce morbidity and mortality in patients with heart failure, even severe heart failure. They also recognized patients with chronic kidney disease, either from diabetes or hypertension, can dramatically reduce proteinuria and progression to end-stage renal disease by ACE inhibitors because of that maladaptive response with increased renin, increased angiotensin 2, causing efferent arterial constriction and pumping up and raising that intraglomerular pressure. So that is the story of ACE inhibitors over the 2 episodes. We're switching gears for the next series, focusing on the different mechanisms of the disease of asthma and those discoveries along the way. Thanks for listening to Mechanism of Action. Give us a review if you can to help other people learn about the podcast. Thanks again for listening.